Good afternoon, dear students. Today we will explore a new topic, general and medical helmintology. Tapeworm sloths, pathogens of echinococcosis, elevococcosis, and diphyllobothriosis. I am Andrei Pavlov, assistant of the Department General and Molecular Biology. This slide presents a lesson plan. First, geographical distribution of human helminthiasis caused by tapeworms. Second, morphology life cycle pathogenic effect of echinococcosis. Third, diagnosis and prevention of echinococcosis. Fourth, morphological features, life cycle, pathogenic effect of elevococcosis, differential diagnosis of echinococcosis and elevococcosis. Fifth, morphophysiological features of the orders of chainworms and sloths. Sixth, morphology life cycle of pathogenic effect of broad tapeworms, diagnosis and prevention of diphyllobothriosis. Helminthiasis is especially diverse and widespread among the population of tropical zones of Asia, Africa, and South America, which is due to the absence of sharp fluctuations in temperature, air humidity, and soil in these areas. This favors the maturation of eggs and larvae of helminths, the development of larval forms of biohelminths in the body of aquatic and terrestrial, intermediate and additional hosts. Echinococcosis, Echinococcus granulosis, the causative agent of echinococcosis. A sexually mature individual with a weight of 0.25 to 0.5 centimeter. Consists of 3 or 4 segments. 1 to young segments, 1 hermaphrodite, 1 mature. The scolex is pear-shaped, has 4 suckers, and proboscis with 36 to 40 hooks. The hermaphrodite segment contains 32 or 40 testers. The yolk is located behind the ovary. The mature segment measuring 2 on 0.6 millimeter contains a sac-like uterus of a closed time with a variable number of lateral branches in which fifth or 800th x. The eggs are morphologically similar to those further tenids, measuring 31 or 40 microns. Thin echinococcal bladder has the appearance of a thick walled bubble filled with a toxic liquid. The inner parenchymal membrane in geminal forms protrusions, blood chambers with scoliosis and child bubbles. The major broad chambers turn scoliosis pour out to the bottom, forming together with small daughter bladders. Echinococcal hydrated scent. Outside of the germ is a thick layered cuticular shell, and then the outer fibrous formed by the host body. The echinococcal vesicle retains the ability to grow root the life of the host. This slide presents an image of the structure of echinococcus on a microscope. The final host is dogs, wolves, jackals, foxes, in which the sexually mature stage of the parasite is located in the small intestine. The intermediate host is herbivores, mammals, humans. Major echinococcus segments break away from the strobila, crawl out of the dog's anus, and crawl on its fur, scattering the eggs. Echinococcus eggs are also excreted in the dog's feces. They remain invasive for 10 months. The invasive stage for humans is the egg. A person becomes infected by ingesting echinococcus eggs with contaminated food and drink from dirty hands when in contact with a sick dog. Herbivores ingest eggs with grass contaminated with dog feces. Oncospheres are released in the small intestine, embedded in the intestinal wall. They spread through the body with the blood flow. Localization in the body of the intermediate host. Liver in lungs, 75%. Muscles, tubular bones, brain, and other organs where echinococcal blisters are formed. The bubbles grow slowly, reaching a diameter of 5 centimeter by the end of the first year. Humans are a biological deed and in the life cycle of echinococcus. Dogs and other final hosts become infected with echinococcosis by eating the entrails of herbivores with echinococcal bladders. Pathogenic effect. Compression of tissue by a growing echinococcal bladder leads to a violation of the function of the affected organ. Dystrophic changes and sclerosis of the surrounding tissues causes a toxic allergic effect when the fluid of the echinococcal bladder is absorbed into the blood. The development of anaphylactic shock and contamination of the pleural of abdominal cavity with daughter echinococcal bladders. In the event of a rupture of a bladder. Clinic. Independent on the location of the bladder, its size, and the immunological reactivity of the patient. In most cases, the disease is asymptomatic and is detected by accident. With the clinically pronounced cause of the disease, the symptoms of compression and the development of the volumetric process of the corresponding organ in combination with allergic manifestations prevail. With liver damage, severity and pain in the right hypochondrium, hepatomegaly, and possible. The blood can be palpated as a painful tumor of a dense consistency with a smooth surface. At a late stage of the disease, compression of the bile ducts, or the inferior vena cava, rupture of the echinococcal bladder that occurs spontaneously or as a result of trauma, separation of a bladder impossible. Diagnostics. Clinical. Epidemiological history, a combination of local origin, damage of things, of allergy, instrumental examination data, radiography, ultrasound, computed tomography, radio sertope scanning, laboratory serological reactions. RSC, RNGA, REM, latex agglutination or reef, skin allergic tests, Katsoni reaction is now rarely used due to the appearance of a group of more sensitive and occurrent serological reactions. Detection of scoliosis and hooks in sputum and duodenal contents when the bladder breaks into the lumen of the bronchi or bile ducts. Diagnostic picture of the echinococcal bladder and microscopy of its contents is possible, but this method is not widely used due to the dangers of contamination by daughter's bladder during the procedure. Prevention. Personal prevention. Compliance with the rules of personal hygiene. Washing vegetables, boiling water. Preventive deworming of domestic dogs twice a year. Public prevention. Destruction of the internal organs of slaughtered animal affected by echinococcosis, extermination of stray dogs, sanitary and educational work. Alveococcosis, Alveococcus multilocularis, the causative agent of alveococcosis. The sexually mature form of echinococcosis and alveococcosis are similar. They are distinguished by the structure of the uterus in mature proglottids in Alveococcus. The uterus in spherical and in echinococcus with lateral outgrowth. The pinna of the alveococcus is filled with gelations, mass, and is a dense knot of small vesicles. Daughter bladders bud only outward in echinococcus inward. Alveococcus is called multicameral echinococcus. This slide presents image of the microscopic photo of Alveococcus. Alveococcus is a typical natural focal disease. The final host wolves, foxes, and Arctic fox with have Alveococcus in their intestines. Intermediate hosts are mouse-like rodents, rarely herbivores, and humans. Alveococcus eggs are released into the external environment with the feces of sick predators. Rodents become infected by eating for contaminated with Alveococcus eggs. In invasive stage of humans is the egg. A person becomes infected through dirty hands when cutting animal skins or eating unwashed wild berries. Localization in the body of an intermediate host and the development of the parasite in the human body are similar of the development of echinococcus. Most often, the liver is primarily affected, then blisters form in other organs, lungs, kidneys, spleen, brain. A person is a biological deed, and in the life cycle of Alveococcus, predators become infected by eating rodents. Pathogenic effect. Toxic and allergic destruction of the tissue of the affected organ. Multiple lesions of internal organs due to the separation of daughter vesicles, and they're spread by the blood flow through the body. Metastasis. Clinic. Alveococcosis clinically resembles the picture of malignant neoplasms of the corresponding localization. Alveococcosis of the Larva occurs as a slow-growing carcinoma and is accompanied by symptoms of hepatic portal hypertension, characterized by hepatosplenomegaly, ascites, jaundice. Diagnostics, clinical, epidemiological history, a combination of local origin damage and scenes of allergy, instrumental examination data, radiography, ultrasound, computed tomography, radioisotope scanning, laboratory biopsy of the affected organ, and microscopy of the obtained samples. Serological reactions. Prevention. Personal prevention. Washing barriers, boiling water. Public prevention. Compliance with the rules of personal hygiene, sanitary and educational work. There are two orders in the class. Idols, Pseudophilidae, and Sepens, Cyclophilidae. Idols. Scolex is equipped with botria. The genital opening open ventrally. Open type uterus. There are three genital opening, one male and two female. Yolk gland follicular. Eggs of the trematode type with a cap. Sepens. The scolex is equipped with suction cups. The sexual opening open on the side of the segment. The uterus is closed. There are two genital openings, one male and one female. A single yolk cell, eggs with a oncosphere covered with a delicate shell. Morphology, a life cycle, pathogenic effect of broad idler. Sexually mature individual 3 to 10 meters long, described worms up to 20 meters long. Consists of 2,000 segments. The scolex is oval with two suckling slits botria, which are located on the dorsal and ventral sides. The neck is thin, 4 or 10 millimeter long. The width of the hermaphrodite segments 10 or 20 millimeter significantly exceeds the late 2 or 4 millimeters. In the back of the body, the segments the square. In the center of each segment is the rosette-shaped uterus. Fluke type X up to 75 microns long. Fin is a worm-shaped plerocercoid with two botria and at the interior end. The main hosts are humans and carnivores, mammals, cats, dogs, Arctic foxes, bears. The first intermediate host is crustaceans, cyclops, diatomus, daphnia. This second intermediate host is fish, and the reservoir host is predatory fish. The eggs of the parasite and released from the body or the final host with feces. For further development, they fall into the water, where after 3 or 5 weeks the larva Coricidium covered with cilia and equipped with 6 hooks comes out. Coricidium is ingested by the first intermediate host. In the intestine, the crustacean Coricidium turns into a procercoid larva, which has an elongated shape and is equipped with 6 hooks on the back end of the body. When the crustacean is swallowed by a fish, in its muscles and genitals, the procercoid turns into a plerocercoid. Predatory fish, pikes and others can be reservoir hosts signs when they swallow the affected fish, the plerocercoids pass into the muscles and sexual system. Pathogenic effect. Mechanical damage the intestinal mucus when it's pinched by botria. Toxic, allergic, poisoning, of the body with the waste products. Nutrient at the expense of the host body and metabolic disorders. Absorption of nutrients, vitamin B12, which leads to the development of malignant anemia. Typical symptoms: general weakness, poor appetite, nausea, abdominal pain and rumbling, flatulence, intestinal disorders, subfebrile body, temperature. There are signs of anemia: sharp general weakness, drowsiness, dizziness, dyspeptic phenomena. Bright red spots or cracks appear on the trachea and atrophy of the papillae of the trachea occurs. The skin is pale with a yellow tinge. The liver and spleen and are enlarged. With a large number of helminths, dynamic of mechanical intestinal obstruction may occur. Diagnosis: clinical isolation of segments with feces. Laboratory: horoscopy of feces. Sometimes you can find parts of the strobila, helmintoscopy. Prevention, personal prevention, good heat treatment of fish, public identification and treatment of patients, prevention of fecal contamination of water bodies.